All right, we are going to review DNA structure. Here is our DNA molecule. You'll notice that DNA looks very similar to a twisted ladder. What's important is that DNA is made up of the building block called a nucleotide. So essentially, this is a nucleotide, which is what is squared off in the picture. And the nucleotide has three different parts. It has a base, a sugar, and a phosphate group. You'll notice that the sides of the ladder, or the backbone, those are made up of alternating phosphate and sugar molecules. You'll also notice that on the inside of the ladder, where the rungs would be located, those are where you'll find your bases. Now, a really important thing about the bases is that there are certain complementary base pairs. So the A base will always bond with the T base, and the G base will always bond with the C base. So we call these bases adenine, thymine, guanine, and cytosine. So adenine always bonds with thymine, and guanine always bonds with cytosine. A couple easy ways to remember these base pairings. One of them is the saying apples and trees, cars and garages. Another way to remember it is that the straight lined letters go together and the curved lined letters go together. These bases are always bonded together by a hydrogen bond down the center of the DNA molecule. So as far as DNA replication, this process is essentially DNA making a copy of itself. That's what replication means, is to make a copy. So in the process of DNA replication, we are taking a single parent DNA and making two DNA daughter molecules. So the steps for DNA replication, the first one is the DNA has to unwind. So DNA is very tightly bound together. It has to get a little bit looser for us to make a copy. So it's going to unwind and unzip. Once the DNA is unzipped, that's when we have all of our bases exposed. And this is when the free-floating nucleotides, so the building blocks of DNA, they can start coming in and pairing up with those exposed bases. Again, A's always bond with T's and G's always bond with C's. Once those free-floating nucleotides start bonding with the bases, that's when we start creating two new DNA daughter molecules. You'll notice that in our DNA daughter molecules, they have one light strand and one dark strand. So what that means is that each of my daughter molecules will always have one strand of the original parent DNA and one strand of new DNA. And that's called semi-conservative. Another process that we need to talk about is protein synthesis. So synthesis means to make, and essentially protein synthesis is the making of a protein. What's really fascinating about DNA is that it's the blueprint of life, which means that it has this special coding to make different proteins that our bodies need. And so we're going to talk about two different steps in the process of protein synthesis: transcription and translation. So starting with transcription, this process happens in the nucleus because that is where your DNA is located. So right here is my double-stranded DNA in the nucleus. We are going to have this special molecule called mRNA come in and actually write down the code in the DNA. So what's going to happen is my DNA is going to open up, and the mRNA, which stands for messenger RNA, is going to go in and basically write down the coding in the DNA. They're basically transcribing the message. If you know what a transcriber does, is they essentially just write down word for word what is being said. And so this is going to be my transcriber, the mRNA, and that's going to write down the message. Now, the message needs to get to a ribosome because that's where proteins are actually made. And so once the mRNA has written down the message, it's going to send that message out of the nucleus and find a ribosome, a protein making factory. Once the mRNA finds the ribosome, then we can start the second process, which is translation. So, translation is essentially what it sounds like. They are going to translate the code from the mRNA. And so, in translation, we have these molecules called tRNA that come in and are specifically able to translate the code into a protein.